Right. For a couple of lectures, I'd like to sort of take a side trip and away from at least talking about modes of inheritance and talk about meiosis and mitosis. I know I can see through the screen that your eyes are rolling back and you're saying, why in the world are you doing this again? I've learned all this, mitosis and meiosis. I know all about prophase, metaphase, anaphase, telophase. That's fine. I'm sure you do. You've had it. Some of the first things you learn in biology, whether it's in high school or general biology, you certainly go through it. What I want to do here, though, is I want to connect it. So I'll ask you a question. If Can you tell me in meiosis or mitosis where Mendel's first principle, how that happens? He was describing something that was going on here. And if you can do that, can you describe Mendel's second principle? Can you illustrate those? If you can, you don't need to watch these lectures. If you can't, then you need to pay attention, because I'm not going to go through here and make you memorize meiosis and mitosis. I assume you know that. What I'm going to do is talk about how they relate and why in the world we've made you over and over again go back and learn about these processes. At least part of the reasons for that. Okay, so part of the reason for us is that, remember way back in introductory lectures, we talked about the fact that the primary model that we think happens as far as reproduction and passing information on is epigenesis. This idea that you get this DNA that's a template, that's a blueprint, and that somehow we manufacture organisms based on that blueprint. Well, those organisms are produced, of course, by this growth and maintenance process, as well as then, of course, once they are grown and become adults, they have to propagate. The growth and maintenance process really involves mitosis. Okay, mitosis, where we are simply taking cells and we are replicating those cells and then potentially maturing them to become, skin cells or colloidal cells or neural cells or whatever they are. These are called somatic tissues. A very important word. A lot of people skip over that word, but somatic means the majority of cells in your body, 99.99% of the cells in your body are somatic, and they're going through mitosis if they're still replicating. Uh, and certainly skin cells, the lining of the intestines, all the other things are somatic cells. Okay, so mitosis happens there when cells get too big, when they're going to form, etc. Now, as far as propagation or reproduction, it can happen through mitosis as well. We've all seen this idea of clones. You've probably experimented with a little hydra or something, and when you're using a microscope and you see the little hydra here, and then you have these buds come off the side, and the bud breaks off, and that becomes a new hydra. It does that by mitosis. These are mitotic cells that are just producing a new organism that then clones itself off. And certainly mitosis is critical for propagation of many plants, strawberries, and all sorts of things are propagated by mitotic event. We take a cutting and then we just simply take those. Those become clones, right? There's no genetic exchange. There's no new types of information that occur there. It's simply taking the existing cells and just making multiple copies of them. Now, we're interested in what involves meiosis, however, okay, propagation by this. And this normally occurs through what are known as germ cell lines. Now, not germs in the common usage of it, which we mean some things that pathogens that attack you, but germatic tissue. Germatic tissue is this tissue that produces in plants pollen or ova, in mammals and vertebrates produces egg and sperm. Okay, these are germatic tissue, is what it's called. Meiosis is responsible for that. It's also where the processes that Mendel was describing actually are put together. Okay, that's where they occur. So let's go back, since it's been a few weeks since we looked at this, and talk a little bit about a couple of things. Okay, just to remind you, a question I always ask, because I say, when does replication occur? When in mitosis and meiosis does replication occur? Okay, in other words, when are sister chromatids produced? Is it in prophase, metaphase, anaphase, and telophase? And hopefully, now that you've seen the replication lecture, you know that none of these are correct. Okay? Replication occurs long before you either get to meiosis or mitosis. Okay? Now, why am I bringing this up? Because there's a term that's associated with this that I just dislike. And that term is called interphase. And we say replication occurs during interphase. Okay? And I really don't like the term interphase because students are really have a propensity to link interphase in with meiosis or mitosis. And they'll say, okay, there's interphase, prophase, metaphase, and somehow it's all linked together, and it has nothing to do with it. Okay, in fact, it's just a really bad term. Interphase really is talking about this whole life cycle of the cell that's going on out here, right? The reason it exists is that for much of the last century, we didn't have really good microscopes, and we would be using these kind of old microscopes, and only time we could see chromosomes was when they were fully condensed up here in prophase, right? Now, the rest of the time, we kind of could see DNA, but not really. We didn't know what the chromosomes looked like because remember, DNA is all stretched out. We talked about the condensation of DNA, beads on a string and all that. It's really, except maybe in late G2, even hard to see it. So we really didn't know what was going on out here. And we just called it interphase. And I think that's really sad. I mean, you know, it's like going to graduation, right? We go out there and we have these little processes, and kind of like mitosis is like that, right? In graduation, you have a processional in, you have some speeches, and then you walk across the stage and shake the provost or president's hand and get your diploma. Then you throw your hats in the air. There's all those steps, but that's not college, right? And no more than interphase is the life of the cell. I mean, you spend a lot of time in college doing other things. So just to say it's inter-college is kind of a low bit. So anyway, that's my little soapbox on interphase. I don't like the term because it really doesn't represent most of the life of the cell. Okay, it's just a lumping, and primarily because we didn't have good ways to describe what's going on, which we now do, right? So, big point. S phase, that's where replication occurs. It has nothing to do with meiosis and mitosis. It can be hours away, days away, years away from the actual end of meiosis or mitosis, okay? Now, usually associated with mitosis is fairly close because we don't spend a lot of time in G2, but we do spend some time there, so it's not a part of it, okay? All right. Now, just to go back, remember that we're talking about having these structures called homologs, DNA that's there before replication. We call them homologous chromosomes or homologs for short. Post-replication, you still have homologs. We now call them a dyad. They're perfect copies. We talked about replication, so we know they're perfect copies. Whatever locus was on one of the sister chromatids over here is found over here, so we should have little a joined up. That's what we have, and joined by some element that's we call a centromere, a dense region of connectivity. Okay, that's what we have. Now, it doesn't matter whether we're going to go into meiosis or mitosis, that's what we have, right? So we start to enter mitosis or meiosis, we go through this reduction phase where we compress during G2, keep compressing. Finally, lamin and other structures come in to give us this sort of skeleton that we can build on. And what people think of when they first start in genetics or anywhere else, that's what a chromosome looks like. Yeah, it does look like that right there in early meiosis or mitosis. Most of the time in the cell cycle, that's not what it looks like. Okay, but we now know that. All right. So we've got all this condensation going on. We know we've got this process that leads us to these function of these chromosomes. So here's where we are. We've got our chromosomes, right? We're pretty close to being in this phase right here. All right. So let me talk about mitosis. I'm just going to briefly bust through it. I'm not going to spend a lot of time. You can go back and look at it if you want, but it's PMAT, prophase, metaphase, anaphase, telophase, as far as we're concerned. We call them Bob, George, Sally, and Sue. I don't care what we call them, except we have to be able to communicate. But traditionally, they're known as prophase, metaphase, anaphase, and telophase. Genetically, what's important is that we started out with a 2N organism. We went through replication. We doubled the amount of DNA. So we're going to call that 4C now instead of 4N because it's really not a good representation of 4N. Okay. During G2, we sit there with a 4C product. We do this final condensation. We put out some messages that we need into the cell to get it ready. We do a lot of production of things to do the process. Then in mitosis, we take that and we break that 4C by mitosis back into 2 cells, daughter cells, each being 2N. That's really all we can do, okay? And we do it through the process of PMAT, prophase, metaphase, anaphase, and telophase. Prophase, shortening and thickening, final shortening and thickening of the chromosomes, the dyads like this, okay? Probably have seen it illustrated where this is DNA in maybe G1 or G2. Okay, that's what it does. Eventually, though, they start to shorten and thicken. By the time you get to prophase, you can actually see them, right? So these are chromosomes as they might appear certainly in early prophase, if not late. Okay, metaphase, middle phase, we line up along some equatorial plane, and we're going to put them, up and down, but it's three-dimensional object like a big balloon, so they could be anywhere, but they're lined up along a plane. There are lots of cool things that go on there that you'll learn about in Cell and other places about the structuring. But for our purposes, we make these little bungee cord-like structures that go out attached to the centromere. These are called spindle fibers. The spindle fibers attach to the centromeres from both sides. And we now have everything lined up in what we call metaphase. So our cells would look something like this, with all of our chromosomes sort of lined up down the middle and little connecting rods, little bungee cords going off to each side. Anaphase is a tearing phase. This is where we do the separation of a 4C to these two ends. We break apart these sister chromatids, and now we have chromatid 1 over here, and say this is a maternally derived, originally homolog from chromatid number 1, and this is the paternally derived chromatid number 1. So we now got potential to have cells that have exactly what you're supposed to have. We've copied them, but now we've broken them apart so that they each get what they want. Now, notice that there's none of this. We didn't take our dyads and put them together. We didn't do any things. In fact, if we were to go back for just a second, when we form in metaphase, there's no relationship between these chromosomes. This could be chromosome pair. This could be dyad for chromosome number 1, and the other one for number 1 might be off down here somewhere. They don't line up 1, 1, 2, 2, 3, 3. They just line up along the plane. Okay, so they have no relationship to each other. The homolog from mom replicates, homolog from dad replicates, but they don't come together in any way during mitosis. So we have anaphase, we separate them apart, they fall apart even further, and then eventually we get them fully apart, something like this. Okay, you've all seen pictures of how it looks with them pulled apart, usually at the centromere, so they form some kind of little V-type form as they go out. Then the tearing phase, which is telophase, which is just finishing the product up, and we end up with 2 cells now, daughter cells, about half the size of the original, but containing exactly the same amount of 2N information. It's got 1 homolog for mom, 1 homolog for dad for every pair of chromosomes that you have. Okay, back to a 2N cell. This is the telophase part. That's mitosis. That's what it's about. Now this cell on the right over here could go off and become a cuboidal cell for intestinal lining or whatever it's going to be. Okay, next time this one might divide again, produce more cells if it's a basal cell, and this cell just keeps going off and maturing along its pathways. Okay, that's what we have. Now, notice that Mendel had nothing to do with this, right? This is all a part of epigenesis. Once we've got this gametes fused together, we're just doing mitosis to make livers and hearts and all the new stuff we got. It didn't tell us how we got this particular homolog versus this one. That's what comes in with meiosis. And that's what we'll look at next. We'll next move to meiosis.